The last decade in biotechnology has been intense. Well, gosh, let's just say the last 5 years, or let's just say the last few months. It's all intense, isn't it? With the global pandemic, the rise of artificial intelligence and breakthroughs that once sounded like science fiction are suddenly becoming real.
You said the last decade, and I thought that seems like a long time. And then I realized it's not actually that long, and now I feel really old. But anyway, the field is moving super fast, and one of the fields that is moving superest fastest is gene and cell therapy. I'm going to patent that, superest fastest. That's a new adjective.
I like it.
Technologies that could allow doctors to treat diseases by delivering genetic instructions directly to the cells that need them. Some of these on the market already, and they are moving at pace.
It sounds simple, but in reality, getting the right genetic treatment to the right cell at the right time in the right organ is a huge scientific challenge.
Yes, it's like just how do you pinpoint exactly where you've got your medicine to go? So in this episode, we're taking a big step back and we're asking a big question. Where does the science actually stand right now?
And to help us explore that, basically, we brought together two experts working at the cutting edge of gene therapy technologies.
We are trying to use those vectors as a good postman to deliver cargo into our brain.
It opens doors to really deep disease understanding and pairing with all those advanced treatment options.
And that's what matters on today's episode of Discovery Matters.
Dr. Lijia Ma is a scientist based in Westlake in China who develops new gene therapy technologies to help doctors send genetic treatments directly to specific cells into a patient's body.
It's a beautiful city, and our university is relatively new to the world. It's about 10 years old.
And I've been to Westlake, and it is a beautiful city, and it is an amazing university, and I just loved hanging out there with the team. So I didn't unfortunately get to meet Dr. Ma. So what does she do there?
What I am doing, actually, my laboratory doing in the Westlake University and my startup called Westlake Gene Tech, we are focusing on gene and cell therapy. Especially, we focusing on how to make gene and cell therapy more accessible to broad populations, which means we cannot make gene therapy drugs that are super expensive. Otherwise, people cannot get it.
So we asked Dr. Ma to take a step back and look at where we are post-COVID, post the emergence of AI and all these things that have happened in recent history, and simply ask, what is the state of science in the world at this moment?
It's a quite broad topic. I think one of the major breakthroughs recently in the entire gene and cell therapy field is that we are trying to reroute the viral vectors we used before, even those non-viral vectors, reroute them inside our body so that they can go hopefully wherever we want them to go.
The major hurdle in cell and gene therapy used to be that most of the vectors, non-viral as well as viral, well, they really liked to go to the liver.
But actually, we have many other destinations inside our body. We really want those vectors to go to deliver effectively, efficiently of the therapeutic payloads. I think this kind of revolution comes from many engineering perspectives. I think the major breakthrough at this moment is that people started to understand how we are going to, if we are engineering the viral vectors, for example, the AAV vectors, by changing a couple of amino acids in the surface of the virus, then what kind of destination the virus vector will go exactly inside the body. I think the human being, just started to establish that kind of knowledge in the past 5 to 10 years. And currently, we started to gain more and more knowledge on that side.
A little refresher here then for folks. Ex vivo cell therapy is, of course, that cell therapy that's happening outside the body. So you take the cells out of the body, blood cells, whatever it is, you re-engineer them, you re-inject them, and then in vivo, which is the new modality du jour, happens inside the body. So you send the genetic instructions to the cells by injecting them into the body, and the cells themselves make the CAR T therapy.
I think I'd like to step back a little bit. Think about that even the ex vivo technologies, the ex vivo CAR T or traditional CAR T, even we say they are traditional CAR T. You cannot imagine it's about like 20 years old. We call them old now because the technology really, developed it so rapidly. So I think for the in vivo part, the biggest hurdle currently is that how we are going to precisely to target a tissue, an organ, or even a specific cell types by using a blind vehicle, the viral vector or the non-viral vector. Typically, we do those deliveries. They are like delivery men, the people who deliver package for different places.
We really went far with this analogy to the postmen, as she called them. And she explained that they're only familiar with a few places in the local area.
They only know a few zip codes. That's an issue. So in order to make those postmen to know a lot of zip codes, we need to refresh their memories. And the biggest thing we need to do currently is that how we are going to engineer those factors to make those postmen to recognize other zip codes.
I really like the analogy. So these postmen, they don't just need the number of the house. They need to know that Conor McKechnie lives in this particular house and that he prefers to get his post at 11:30 in the morning.
Oh, you have a nice, luxurious morning, Ben. I like to get my post much earlier, but essentially, sure. What you said there about the specific time of day that the postman would deliver your post, Dr. Ma says that is an essential part of the whole process here.
It's definitely a very critical level of, we are talking about precise in vivo gene delivery. But we also have another level of controlling we really want to achieve is that about the map. What we are trying to do is that how we are going to refresh the postman's memory so that they can get the map of the entire community comprehensively. And they know exactly, say, okay, 2 or 3 p.m. afternoon would be a good time to deliver stuff for Dodie, right?
And Dodie gets her post at 3 in the afternoon. I mean, come on.
It's not my analogy, but I like it. I think it's a good one. And I thought it was such a good analogy that we also asked a colleague of ours, Peiqing Zhang, scientific director at Cytiva in the genomic medicine business, to jump in on the action.
So, imagine now we have a well-trained postman who memorize, let's say, 1,000 locations and the timing of the delivery. So that's fantastic. We want to have now 100 postmen to do the job. And how do we scale the training to make sure each of them knows exactly where to go and what time to send the delivery? So that is, analogy, a biomanufacturing challenge, isn't it? So imagine now we actually work out the design phase of making AAV precise, safe, and powerful. In doing so, to achieve so, we must stack up many layers of design elements. In biology language, it could be, let's say, time-sensitive inducible promoters, or it could be some sort of biological triggers.
All these components must be organically synthesized together into the virus particles,
so that they can carry those biological properties. Now, translating to biomanufacturing, each one additional layer of biological design element is going to level up the biomanufacturing challenge, right? So just to give you a few examples, AAV is not known for being able to package in large cargoes, right? So as we increase those design elements, how do we ensure the AAV capsid is still able to still squeeze in that large memory, right, so to speak? And how do we make sure it can consistently produce AAV of comparable product quality batch to batch, and also in a cost-effective manner. As much as we are getting really close to some design principles and design answers, the biomanufacturing front are still full of challenges. But again, we are also seeing light out of the tunnel in terms of biomanufacturing.
I think that aspect of it is funny. I love it, but it's ironic that the closer you get to that fine detail of personalization, the more you need to be able to scale.
Mm-hmm.
And so here we go. I feel 2 letters marching with inevitability towards this sentence.
The one is right there at the start of the alphabet.
And the other one, yes.
Is I.
There's no team in I. Oh, it is, of course, AI.
And here is Dr. Lijia Ma again.
The issue is that we just talking about the scale of the data sets or the search space. We needed to try every single possibilities to identify the real zip code that the postman need to go. And the reality is that we cannot experimentally let the postman to try every single route to memorize the where to go. So what scientists are doing currently is that we do a small portion of experiments from the unlimited search space. Then by sampling that small amount of routes, and then we train the postman, say, okay, you just do these routes one every single time. Say if you trained 1,000, 10,000, 100,000, 1 million times, then they will be able to sort of understand what was the best way to a zip code. He or she never been there. We are facing to a problem of unlimited search space for postmen. And because of the limitation of the current experimental scalability, so we just let the postman to do a small portion of the experiment, do assembly, AI training. Hopefully, he or she can understand the rest part of the space. He or she never been there. Yeah, that's where AI can help us in terms of to develop a better in vivo delivery vector with a good memory, knows where to go, when to go.
Fascinating stuff. What does Dr. Mark think she is going to accomplish, not in 5 years or 10 years, because that's always, where we think that the boundaries of medical science are. But what about now, in the immediate future?
We focus on peptide engineering for a few years. At the beginning, we figured out the example I just made, the AI kind of thing to train postman would be a good way to let the postman to go the places he or she never been there. Then the second question we faced a few years ago is that what kind of destinations we actually want the postman to go?
We need some more specific examples.
So the by default destination the postman knows is the liver. Everyone's body. The liver is the best, is the most familiar places, and every single postman knows to go. But actually, there are many places, many destinations that we really want to deliver the therapeutic payloads there. For example, brain. For example, muscles and other organs like heart. We are making brain as an example. Brain has some really unique features. It's relatively immune privileged because our body needs to protect our brain to prevent the external infection to get in. So there is a tight structure called blood-brain barrier. In short, we call it BBB, right? Blood-brain barrier is doing a very good job in terms of protecting our body from infections, any kind of infections, including viruses. When blood-brain barrier is helping to protect our body from infection, it also pre-excludes the infection of those delivery vector, which we are trying to use those vector as a good postman to deliver cargo into our brain. So that is one of the destinations we really want the vector to go.
So before we finish up, and in order to sum up, I would like to hear from Peiqing again. What does he think people should know about advances in the science and how we are helping our bodies to understand how to heal themselves?
I guess the keyword that I thought of is really options. I think science or the advancement in science is really providing more options to people. Wide access to whole genome sequencing, I think you really opened doors to a lot of deep disease understanding, even at the individual level. Nowadays, you can do a swab and send it for your personal whole genome sequencing at the cost of maybe $10 or $30, right? So then you can really understand a lot of things, maybe your genetic susceptibility to certain diseases, or even your ancestry, right? Where you actually came from, or your family tree actually came from. But I think deeper than that is really, I think it opens doors to really deep disease understanding and pairing with all those advanced treatment options, right? So we talked about viral vectors, we talked about non-viral vectors, ex vivo, in vivo. We are not saying that one will certainly win over all the rest. We are saying that it really depends on the diseases, it depends on the situation. They will find their own places, right? So I do believe the future is going to be the combination and also the convergence of all those different modalities, offering options to patients. Options, that is what I would proudly share with my neighbors, what modern biological science is going to bring.
It's just brilliant to hear that so much of the future of medicine comes down to something as relatively, here in the air quotes, simple and fundamental, close air quotes, as delivery.
Sure, the therapies themselves, there's so much optimism about them. They are powerful, but if you cannot get them to the right cells at the right time, they simply will not work.
And it's just clear from both of them that it's just a super complex challenge. You're not designing a therapy, you're designing a whole biological delivery system, and it has to work in concert with what's going on in the body already. It's just incredible.
That's right. With engineering viral vectors, understanding the biology of different organs, and increasingly using AI to navigate an enormous design space.
And I think this is where the analogy of the postman really breaks down, because my postman walks the same route every day, day in, day out, come rain or shine, in shorts as well, all the way through the winter. But this is a completely different navigation challenge, right? They're not going to the same place all the time. They've got to get to exactly the right spot, and that spot changes with every disease.
That's right.
So
let's get our homework out.
Let's do that. Every day is a school day. What have we learned this week?
You go first.
Okay, so this is super fun. At the time of recording, which is kind of towards the end of March 2026, something really hilarious, AI again, has been breaking in the news. And this is a story around the fact that the drivers of the future of biological therapies may not actually be the biologists. It may be the people who have the skills and capabilities on the big data and data engineering side and the AI side. So this week, we came across the news story that ChatGPT has been used to design a cancer vaccine. And the first patient was, you're going to like this, Dodie, a dog. And the story comes out of Australia, where a tech entrepreneur, essentially, whose dog was diagnosed with an aggressive cancer, decided not to accept there's nothing more that we can do. And he used generative AI, including ChatGPT, as a scientific co-pilot. And he wasn't asking ChatGPT to treat the dog. What he did was he used it to help him design a personalized mRNA cancer vaccine, and the same underlying technology that we now associate with COVID vaccines, but aimed at the cancer instead of being aimed at the flu virus, for example. And it's brilliant. So you sequence the tumor, you identify the mutations unique to that cancer, you encode those targets into an mRNA vaccine, you stick it in the dog, and bingo.
And how is the dog?
The dog is cured. ChatGPT sped up the design process, synthesized literature, checked assumptions, stress tested all the ideas that would normally take weeks and months, and the cancer went into remission. It's not proof, it's not a clinical trial, and it's not a shortcut.
It's one.
Yeah. But, just you can teach an old dog's
Oh,
immune system new
hilarious.
tricks.
Oh,
Get that?
okay.
I like
All
that
right.
one.
Okay, so that's a nice bridge because my story is also fun. It also has to do with tech, and it's also incredibly silly and will appeal to any seventh grader's sense of humor. So you know what IoT is, right? The Internet of Things.
Internet of Things. Yeah, everything's connected.
Everything is connected. So this story from Science Daily, which is where to get all kinds of good, weird science stories, has a story about smart underwear. So connected underwear. And scientists have put sensors on this underwear to detect, well, quite simply, farts.
Oh,
That's
I knew it was going to be farts.
It's a fart story. That's right.
Oh, I know.
But here it is. And this is more scientifically, the underwear are detecting hydrogen gas produced by gut microbes. So humans who are wearing these connected underwear, and I don't know what the control group was, but humans apparently pass gas about 30 times a day. And this
Not
is
me.
More, Connor?
It's just, I don't do that.
All right.
I don't do that.
It's antisocial. Okay, but here's the real scientific purpose of these connected underwear is to get non-invasive data on gut health and digestion and disorders like irritable bowel syndrome. So that's the serious attempt to turn farts into decent medical insight.
Okay, that's amazing. So this goes beyond the you smelt it, dealt it, because... Okay, we have to stop.
Let's hope that our sound magician, Tom Henley, finds a place for a few sound effects there.
Oh, dearie me. Oh, dearie me.
Speaking of Tom Henley, he is our producer and mixer. And my name is Dodie Axelson.
And my name is Conor McKechnie. Make sure you rate us on Spotify or whichever platform you use to listen to Discovery Matters. Thank you. It is brought to you by the nice people at Cytiva.